So I wanted to create a video to walk you guys through how to determine or identify the phase of meiosis. So what I'm going to do here is I'm going to highlight some pictures of some cells that you've seen before, but kind of highlight the things that I would look at to figure out what stage of meiosis we're in. We're going to begin with this picture. So first of all, when I look at this picture, I'm right away noticing that the sister chromatids have been split and are being pulled in opposite directions. So that's one piece of information. When we look, I can tell here that there are two cells present. If I was going to describe these cells as haploid or diploid, I would say that these cells are haploid because they have only one copy of each chromosome. It's a little hard to tell here because the chromosomes aren't color-coded. That's what we're dealing with. Examining these cells further, when we have two cells, the chromatids are being pulled apart. You could tell they were lined up in the middle, but now they're working towards opposite ends of the cell. So I'm going to pause. You can say what you think this is, and then I'll identify it. All right, so this is anaphase 2. So the chromosomes moving apart is the key part for identifying that it's anaphase. These, remember, were copies. They were identical copies on the left and on the right side, each chromatid. And now they're moving towards the opposite ends. We're going to end up with 4 daughter cells here eventually. We know it's anaphase 2 because the first round of cells is already split. Okay? If it was anaphase 1, the chromosomes would have been present as pairs, as tetrads, and we don't see that here. The next phase up, really key, number one thing we're going to look at right away is here in the nucleus. We see loose DNA. It's uncoiled. We're going to call that chromatin, and right away that should identify what stage we are in here. So I'm going to pause. Good. So this is interphase. All right, with the loose uncoiled DNA and the solid nuclear membrane tell us that this is a cell in interphase. So looking at these two cells, obviously they've been labeled haploid, so we're not going to ignore that. But once again, we have 1, 2 cells, so that should be giving you a hint. We can see that the chromosomes are still in their replicated form with the X-shaped structure. Looking at some other features in these cells that are interesting, up here we've got centrioles and spindle fibers forming. We have a nucleus that has large holes developing, gaps, so the nuclear membrane is going to break down. Give you a second. Good, so this should be prophase 2. Prophase 2 is when the cell is preparing to undergo the second round of division. Okay, so look at here, we've got one cell, we've got replicated chromosomes are still being held together. They are, however, being moved away from each other towards opposite sides of the cell. So this is going to be anaphase 1. It's anaphase 1 because the cell hasn't yet divided into 2 cells. It's anaphase because the chromosomes, the replicated chromosomes, are being pulled away from one another. Okay, so let's look at this cell. First thing we should notice is that it's still just one cell. The membrane hasn't divided yet, but we can see that the membrane is starting to pinch in. Another very important detail in this cell is the nuclear membrane is reforming. Okay, also the chromosomes aren't really organized at all here. They still are in their X shapes, so the replicated chromosomes are still together, but they're not in a line or anything like that. So that would make this cell, it's about to form 2 nuclei, telophase 1. There's a big hint here. First up, we see we've got 1, 2 cells. Also, huge hint, chromosomes are in a straight line down the middle of the cell, and they're not in the tetrad anymore, so you don't have two X's next to each other. Okay, so that tells you we've got to be in meiosis 2. The fact that they're in a line straight down the middle tells us that this is metaphase 2. All right, so here we just have one cell. Very importantly, the homologous chromosomes are lined up next to each other so that they're in, remember we call this the tetrad, with the mom's chromosome and the dad's chromosome overlapping, and that only happens during really just during 2 phases of meiosis, so that should be a big hint to you. The nucleus is still, the nuclear membrane is still breaking down. The spindle fibers and centrioles are still moving towards opposite ends. That should tell you a lot. So the tetrads, the nuclear membrane breaking down, this is prophase 1. Okay, we're getting close to the end here. We have 2 cells, 1, 2, but notice that the membranes are starting to pinch in, and we're getting 2 nuclei in each cell. So a total of 4 nuclei are going to be formed here because it's 2 nuclei in each cell, 4 total nuclei. Because we don't have X-shaped chromosomes anymore, we know that we're in the second round of division for meiosis. This is telophase 2. So last up, these chromosomes are lined up right down the middle, but it's the tetrad, so it's the homologous chromosomes with basically the dad's copy on one side and the mom's copy on the other side. Okay, and it can be reversed over here, so it could be red over on this side and blue on the other. Okay, this is a big clue, so that tetrads have not yet been separated, so we've got to be in meiosis 1 because they're lined up in the middle. This is our last one, metaphase 1.